This is Mazen Judi, Assistant Professor in the Baghdad Medical College in the Medicine Department. I would like to present this lecture to the third stage as first lecture in the medicine, and it's about the infection as introduction of infection and diagnostic methods of infectious diseases. The contents for this lecture include definition of infection, infectious agents, classification and the important infectious agents to the human being, the term normal flora, and discussion about the host pathogen interaction, the method of transmission of infection, together with diagnosis of infectious disease. Regarding the infection is defined as the establishment of foreign organisms or infectious agents in or on a host, which is a human host. Yes. Actually, the host pathogen interaction is dynamic and complex. Whilst it's rarely in the microorganism's interest to kill the host on which it relies for nutrition and protection, the manifestation of disease may aid its dissemination. That's to say, the patient may develop diarrhea and sneezing, and by this manifestation there will be spread of the infection. This diagram showing the chain of infection. As we see here, this is the infectious agent, and is the organism that causes the disease, while this is the reservoir. The reservoir is the place where the population of an infectious agent is maintained. It could be human being, could be animal. The portal exit is the point from which the infectious agent leaves the reservoir, and there will be transmission then. There will be transmission of this infection, which is the process by which the infectious agent is transferred from the reservoir. Okay? So it is the way by which the infectious agent will transfer from the reservoir to the human host, either directly or indirectly. And then there will be entry of this microorganism is to the body site that is first accessed by the infectious agent. And finally, there will be, in order for disease to ensue, the person to whom the infectious agent is transmitted must be a susceptible host. So this diagram showing the chain of the infection starting when it is in the reservoir, which is maybe human being or the animal, reaching to the host and causes the disease. Okay. Now, what are the infectious agents? There are different types of infectious agent, and first actually is established by Robert Koch in the 19th century. Now, the infectious agent can be considered being prions or viruses, or what is called prokaryotes, or what is called eukaryotes. Now let's discuss the term prions. Prions are unique amongst infectious agents in that they are devoid of any nucleic acid, the result of accumulation of protein which forms amyloid in the central nervous system, causing a transmissible spongiform encephalopathy. So this type of infection can cause encephalopathy, and sometimes it is implicated in clinical features similar to dementia. The other infectious agents, which are called the viruses, are incapable of independent replication. instead subverting host cellular processes to ensure synthesis of their nucleic acid and proteins. Viruses contain genetic material, which is called the genome, and maybe single or double-strand DNA, RNA, and the retroviruses transcribe their RNA into DNA by reverse transcription. This picture showing the virus, which is coronavirus, characterized by presence of the genetic material, which is genome. It is one of the group of the RNA viruses. Yes. Now, the virus may be enveloped or non-enveloped. Enveloped viruses are less able to survive in the environment and are spread by respiratory, sexual, blood-borne routes, including the arthropod-based transmission, while the non-enveloped viruses survive better in the environment and are predominantly transmitted by the fecal-oral or less often respiratory routes. The other group of infectious agents was called the prokaryotes, which include the bacteria, including the mycobacterium and actinomycetes. The bacteria, generally speaking, can be classified whether being gram-positive or gram-negative bacteria, and there are specific types of bacteria that are not cannot be stained by gram stain, such as the Mycobacterium tuberculosis. Mycobacteria is a group of bacteria that are called acidophilus bacilli that need a specific stain for diagnosis, as we will see later on, what is called Zane-Nielsen stain for the Mycobacterium tuberculosis, and also other group what is called actinomyces. All these are related to the specific group of the microorganisms which are called the prokaryotes. Prokaryote cells are capable of synthesizing their own protein and nucleic acid and are able to reproduce autonomously, although they lack the nucleus. Okay, like this picture which is seen here as prokaryotes group of infectious agent. The eukaryote, like the fungi, protozoa, and helminths, this is another group of infectious agent, and these eukaryotes contain the functional organelles like the nucleic acid, mitochondria, Golgi apparatus. Eukaryotes involved in human infection include the fungi, protozoa, and these are unicellular eukaryotes with a flexible cell membrane. And also another group of eukaryotes that can affect and cause disease to the human being. These are the helminths, which are complex multicellular organisms including the nematode, trematode, and cystodes. Protozoa and helminths are often referred as what? As parasites. Okay, so the term parasite refers usually to the protozoa infection and to the helminth infection. Okay. In addition to the infectious agents that are cause disease to the human being, there are certain microorganisms are called normal flora. Every human is host to a large number of colonizing microorganisms which constitute the normal flora. So these microorganisms live in the human being, but they don't cause the disease. Actually, the human being get benefit from these normal flora. Resident flora are able to survive and replicate at the body sites, whereas transient flora are present only for a short period. So these normal flora could be resident flora or could be transient flora. Maintenance of the normal flora is beneficial to health of the person. For example, lower GI tract bacteria synthesize and excrete vitamins as vitamin K or B12 or as examples, Also, colonization with the normal flora confers colonization resistance to reinfection or to infection with pathogenic organisms by altering the local environment, for example, lowering the pH of the environment, producing antibacterial agents, for example, what is called bacteriocins, also fatty acids and metabolic waste products, and also inducing the host antibodies which cross-react with the pathogenic organisms. So these normal flora are microorganisms live in the body, and the body gets benefit from these flora. Regarding the host-pathogen interaction, the pathogens are the microorganisms that cause disease to the human being. There is interaction between them. The harmful manifestations of infection are determined by a combination of virulence factors of the organisms and the host response to the infection. That's to say, the microorganisms that affect the body has a certain factors that cause the disease, which are called the virulence factors, and these factors are different in activity from microorganism to other microorganism. The harmful manifestation of this pathogenic infection to the human being, it depends on the combination of the virulence factor of the microorganism and also on the organism host response. When the person or is a good immunity, has a good immunity, can defend himself and can act against the microorganisms. When there is immunocompromised condition, for example, patient is diabetic or patient has transplant, organ transplant, or on chemotherapeutic agent for malignancy, this patient called immunocompromised, so his immunity against the infection is low. So the net result will be different from person to person and from infection to infection. Despite the obvious benefit of intact host response, the host is being the human being. So in spite of this intact host response, an excessive response is undesirable. Sometimes excessive response to the infection will lead to hypotension and to other clinical features. A febrile response is a response of human being to infection. Is altered in infectious disease, microbial biogenes. Why there is fever in the infection? Because of microbial biogenes or the endogenous biogenes released during the tissue necrosis stimulate specialized cells such as monocytes, macrophages. These cells release certain substances, and the net result will be certain substances cause alteration in the responsiveness of the thermosensitive neurons in the thermoregulatory center. The thermoregulatory center localized in the hypothalamus, and when there will be infection, there are certain substances called biogenes. These are released as from the microbial substances or the endogenous biogenes. These biogenes will lead to change in the response of the hypothalamic center as thermoregulation, and the patient will develop fever. Also, sometimes the patient develops rigor in addition to the fever that occur when the body inappropriately attempts to reset core temperature to a higher level by stimulating skeletal muscle and other activities. Okay, so as a response of the patient to the microorganism infection, which is pathogenic infection or pathogenic microorganism, is different from person to person according to his immunity, and also different according to the virulence factor of the organism, and the patient may develop fever, may develop other features accordingly. Yes. Now, what are the methods by which there will be transmission of the infection from person to person? Infectious agent may be transmitted by one or more of the following routes. The first route is respiratory route, by inhalation. Inhalation of the droplets from the patient to the healthy human will develop, this healthy person will develop infection. For example, viral infection. For example, influenza viral infection. Fecal-oral route, also ingestion of infectious material originating from the fecal matter. This is an important route for many infectious problems. Sexual transmitted disease, also direct contact between the mucous membranes of the persons, can lead to the transmission of certain infections. Blood-borne infections, direct inoculation of infected blood or body fluid, sometimes a blood transfusion, is a major source of infection, especially the viral infection like hepatitis C virus. Also, another route is direct contact. Very few organisms are capable of causing infection by direct contact with intact skin. So by, for example, by shaking of the hands between persons can be a source of transmission of the infection. Sometimes via a vector bridge the gap between the infected host or reservoir and the uninfected host. Okay, so these are the ways by which there will be transmission of the infection from person to person. Now, how can we diagnose an infectious disease? Of course, as any other disease in the can affect the body, it depends on three major important things, which are the history taken from the patient. All the symptoms that the patient suffer from them should be known in details by the doctor, and also by clinical examination. This is, of course, we take the signs of the disease, and then we do the investigation. Now, what are the investigations that help in the diagnosis of the infectious disease? Investigations in general for the infectious disease include a group of tests. There are what is called general investigation, include the full blood count, plasma C-reactive protein. Of course, in the full blood count, we will have an idea about the white blood cells of the patient, whether the patient has leukocytosis, lymphocytosis, or has leukopenia, and also the hemoglobin, the platelet count. All these details can be known by full blood count. There are non-specific reacting proteins can be elevated also during the infection, like plasma C-reactive protein, procalcitonin, also cell count in the urine, and also in the cerebrospinal fluid. All these investigations are not specific for the infection, but can give an idea about the pathogens that cause the disease. For example, in the general examination, there are a long list of things can be known. In CSF examination, also we know the cells, whether the type of the cells. Also we know the protein content, the glucose, all these non-specific markers of inflammation or infection. The second group of the investigation, these are the investigation to detect direct detection of the organisms or organism components by using microscopy, or detection of organism component through the detection of the antigen that are produced by the organism, or the toxins that are produced by the organisms also. So antigen may be a part of the composition of the microorganism, and the toxin may be product of this microorganism. Also, another way of direct detection is the detection of the nucleic acid amplification, for example by PCR, polymerase chain reaction. Okay, so these methods are methods as direct detection of the organism or organism components. The other method for the diagnosis is the culture of the organism, plus minus antimicrobial sensitivity testing, what is called culture and sensitivity test, as we will see later on. Also, the other test is for the host-specific immune response. It's very important. Antibody detection, interferon gamma release assay, what is called IGRA. These are examples for the test as host-specific immune response. So, different types of investigation can be done for the patient with infectious disease. to reach the final diagnosis. Next. Now, for detection of the whole organism are detected by examination of the biologic fluid or tissue using the microscope. So here, the microscope plays an important role for the diagnosis of the disease. In the light microscope, which is called bright field microscopy, the test sample is interposed between the light source and the objective lens. And here we use stains to enhance the visual contrast between the organism and its background. For example of this test, what is called the Gram stain. Also, we have Zane-Nielsen stain. Other stains, acid-fast bacilli, can be by acid-fast stain, which is Zane-Nielsen stain in tuberculosis. Also, other specific stains. In this picture, for example, we see here the microorganism after we use culture media and specific media. We take then a specimen from this microorganism and put it on the slide and stain it by a gram stain, specific stain. Will give an idea if there is this bacterial infection the cause of the disease. If the bacteria, for example, arranged as elongated in shape and they take the gram stain positive, which appear as bluish in color, like this color, okay, as we see here, which are the gram positive bacilli when it is elongated in shape, called bacilli. Here, example of this bacteria, what is called Clostridia species, okay. Also another species called Cyanobacterium species. While if they are rounded in shape and arranged as a group of clusters, what is called the clusters of these cells, like this picture, and here what is called the gram-positive cocci, for example, Staphylococcus bacteria. Sometimes these are gram-positive, that is to say bluish in color by gram stain, but these arranged in chains, not in clusters, like this picture. Here it is called what? It is called gram-positive cocci in chains, for example, caused by the Streptococcus. So we diagnose the type of the bacteria by this method, which is gram stain for the bacteria. While if the stain of the bacteria in this method is not bluish in color, it take reddish in color, it could be caused by a specific type of bacteria. If they are rounded in shape, what is called the cocci, and these are the gram negative, now we think the etiology could be what is called Neisseria infection, or could be Marxilla infection. While if it is gram stain negative, that's to say it is not bluish, it is reddish in color, but these are arranged shape is bacilli, not rounded. Now we think of the Enterobacteriaceae, another group, okay, like Acinetobacter species in this picture. So by light microscope and the use of specific stain, we can diagnose the causative bacteria for the disease. Regarding the acid-fast bacilli, which is the Mycobacteria tuberculosis, which is one of the common infections in our country, they take specific stain, which is called the Nielsen stain, not in the gram stain. Another type of the microscopic examination, what is called the dark field microscopy, in which light is scattered to make organisms appear bright on the dark background, and is used for diagnosis of specific infection, for example, syphilis. Okay. Another type of microscopic examination by the use of the electron microscope. This may be used to examine the stool or physical fluid to detect the enteric and herpes viruses, but its use has largely been supplanted by nucleic acid detection. Okay, so the main condition for detection of the whole organism by the use of the light microscope and special stain. The other method is detection of component of organisms. Okay, now detection of the component of the organisms detected for diagnostic purposes include the nucleic acid, which is very important, cellular molecule, toxins, and other antigens. One of the important tests is the nucleic acid amplification test, which is abbreviated in NAAT. Specific sequences of microbial DNA and RNA are identified using the nucleic acid primer, which is amplified exponentially by enzymes to generate the multiple copies of the specific sequence. And this method, what is called the polymerase chain reaction, which is abbreviated as PCR, which is used mainly in the detection of many more organisms like the COVID-19 dependable test. The most commonly used amplification method is the polymerase chain reaction. Reverse transcription PCR is used to detect RNA from the RNA viruses, for example hepatitis C virus and HIV. Now, nucleic acid sequencing is also used to assign the microorganisms to specific strains according to their genotype. For example, detection of the MECA gene is used to screen for MRSA. MRSA is a specific group of the Staphylococcus aureus, which is previously discussed as Staphylococcus aureus, as we see here, is a group of gram-positive cocci arranged by gram stain in clusters like this. A specific group of Staphylococcus aureus called MRSA, which is methicillin-resistant Staphylococcus aureus. A nucleic acid amplification test can detect this group of Staphylococcus aureus, and this group needs specific treatment, of course. This amplification test is the most sensitive direct detection method and are particularly useful when a rapid diagnosis is required. They are used widely in the virology, where the possibility of false positive results from colonizing or contaminating organisms is remote, and are applied to blood, respiratory samples, stool, urine, so different specimens can be used for the diagnosis by PCR and amplification test. Another important and very important test for the diagnosis is the culture for the microorganism. The microorganisms can be both detected and further characterized by culture from clinical samples, for example tissue, swab, and body fluid. For example, patient may have chronic pharyngitis and throat infection. We can take without non-causative microorganism. We can take throat swab for culture and sensitivity, urine for culture and sensitivity, and sometimes tissue taken for culture and sensitivity. There are many methods for the culture. Could be in vivo culture, ex vivo culture, and the most important is in vitro culture. In vitro culture is artificial culture media. These are artificial culture media of bacteria and fungi, and is used for definitive identification and for a test for antimicrobial susceptibility. This is the major benefit, and when we culture the microorganisms, also we put antibiotic with this culture media, and we see if this microorganism cause this antibiotic cause destruction of this microorganism, then this microorganism will be sensitive to this antibiotic. That's to say, we put the microorganism in specific artificial media, and we put with it antibiotic, and we see if this antibiotic active against the growth of this microorganism, then this antibiotic can be used for the treatment of this infection. An important thing is regarding the blood culture. A blood culture is used for rapid microbiological diagnosis, which is required for bloodstream infection. If the patient has severe clinical features and the blood culture now is required, has fever, severe clinical features, and the blood culture plays an important role for a diagnosis of this disease. To diagnose the bloodstream infection, a liquid culture media is inoculated. A specific media should be used for the blood culture. With the freshly drawn blood transported to the microbiology laboratory and incubated in a system that monitors it constantly for the products of microbial respiration, mainly CO2, and generally using of fluorescence microscope or fluorescence method. This showing what I said. When we want to detect the bloodstream infection, we use culture media. First, we have to use a very sterilizing method. It should be sterilized the site of aspiration of the blood from the patient, and the specific culture media, which are this media is liquid media, and then transport the samples as quick as possible to the laboratory. There will be incubation of this media for 5 to 7 days for the culture, and then growth of detection of the microorganisms in the culture media. We take a specimen from this microorganism, as we see here, drops of the from the media of the liquid, put on the slides and then examined under microscopy. And also we can use this microorganism in specific culture medias to detect the sensitivity of the microorganism to a specific drug, and then we give the report. So these are the steps for the diagnosis of the blood culture, the use of the blood culture for the diagnosis of microorganism infection. This picture showing the importance of the culture and sensitivity for a group of bacteria, what is called Streptococcus bacteria. In the Streptococcus bacteria, we have two groups, what are called alpha and beta hemolytic Streptococcus. These Streptococci cause hemolysis of the blood. Hemolysis, but the hemolysis is different whether it is partial hemolysis, like in this picture, or whether it is complete hemolysis. If there is partial hemolysis, as is seen here, this goes with the alpha type of the hemolytic Streptococcus. And the organism shows, for example, Streptococcus pneumoniae. And this picture is taken from the CSF of patients with meningitis. Inflammation of the meninges called meningitis, and the suffix itis for every tissue in the body gives an idea of inflammation of that tissue. For example, hepatitis for the liver, meningitis for meninges, and so. So if the patient suffers from features of fever, headache, sometimes neck stiffness, and so, we will suspect in the presence of the meningitis. CSF is aspiration from the patient and can be seen for the culture and sensitivity to diagnose the microorganism that cause this type of meningitis and give the appropriate management. If there is incomplete or partial hemolysis, and there will be suspicion of alpha hemolytic Streptococci, while if there is complete hemolysis, as we see here, then there will be, as we see here, there will be suspicion of the beta hemolytic Streptococci. Okay, so in this condition, the agar, which is the culture media, which is called agar for the bacteria, becomes transparent, while in condition of partial hemolysis, it is about green in color. Okay. Now, what is about the antimicrobial susceptibility test? As I said, we put antibiotic that is in the site of the agar, and we look for the sensitivity of microorganism to this antibiotic. If there is good sensitivity and good response, then it will be this microorganism sensitive to this antibiotic, like in this picture. We put different antibiotics. For example, we put cephalosporin in one of the discs. Other disc we put another fluoroquinolone and other antibiotic. And now we look for the susceptibility of the microorganism to this antibiotic. As we look here, there is zone of inhibition to this disc. So this microorganism in this agar is sensitive to the antibiotic, which can be used for the treatment of this disease. Now, another important group of the tests is specific immunological tests. This depends on the serum of the patient and under the title of serology. The term serology describes the test carried out on the serum of the patient that is used to include both antigen and antibody detection. The serum can be separated from the blood component by specific tests, which centrifugation test. After we centrifuge the blood of the patient, we take the serum, and now we look for two things, either for the antigen or for the antibody. Antigen is the component of the microorganism that causes the disease, while the antibody is the response of the host to the microorganism, which can be also detected by this serological test. So organism-specific antibody detection is applied mainly to the blood. Results are typically expressed as titers, that is the reciprocal of the highest dilution of the serum at which antibody is detectable. For example, detection at serum dilution of 1/64 gives a titer of 64. Okay, so the titer of the result should be will be the result of this test. The seroconversion is defined as either change from negative to positive detection or a fourfold rise in the titer between the acute and convalescent serum samples. This term, seroconversion. Earlier diagnosis can be achieved by detection of the immunoglobulin M type. The antibodies of the body are different. The immunoglobulin IgM, which is immunoglobulin M type, are produced early in the infection. So early diagnosis can be detected by detection of the immunoglobulin M antibody. A limitation of these tests is that antibody production requires a fully functional host immune system, so there may be false negative results in immunocompromised patients. That's to say, this antibody produced by the host, by the human being, the person that is infected by the microorganism, but sometimes the patient has no capability of production of these antibodies, being immunocompromised. For example, a patient that has renal transplantation, patient has AIDS, that is the human immunodeficiency syndrome, or patient may have malignancy, that is cancer. So this patient may be not able to produce antibodies against these microorganisms. The antibody detection tests include different types of the investigation. First of all, what is called ELISA, which is enzyme-linked immunoabsorbance assay. The other one, it is the immunoblot, which is called Western blot. Also, we have CFT, which is complement fixation test. We have agglutination test and other different tests like the immunodiffusion and the immunochromatography. Yes. Regarding the ELISA can be configured in various ways, as we see here. The first way, which is the antibody detection ELISA. In antibody detection ELISA, the patient antibody binds to immobilize specific antigen, as we see here. This is the antigen, and the antibody here binds to immobilize specific antigen and is detected by addition of anti-immunoglobulin enzyme conjugate and chromogenic substrate, which is this. Okay, this is the first type of ELISA, which is antibody detection ELISA. The other type, what is called antibody capture ELISA, and here the patient antibody binds to immobilize immunoglobulin subclass specific antibody, as we see here. Okay, and is detected by addition of a specific antigen followed by antibody enzyme conjugate and chromogenic substrate, which is this. So this is this type, which is called antibody capture ELISA. The third type of ELISA, what is called competitive antibody detection ELISA, and this group, patient antibody and antibody enzyme conjugate bind to immobilize specific antigen, like it is seen here. Okay, the other group, what is called the double antibody sandwich ELISA for antigen detection. The patient antigen here binds to immobilize antibody and is detected by addition of antibody enzyme conjugate and chromogene substrate, as we see here. This is the antigen of the patient. Okay, so generally speaking, we have 4 types of ELISA for the diagnosis of infectious disease. Could be antibody detection ELISA, could be antibody capture ELISA, could be competitive antibody detection ELISA, and could be double antibody sandwich ELISA for antigen detection. Yes. The other test, what is called the immunoblot test. The microbial proteins are separated according to the molecular weight by polyacrylamide gel electrophoresis, which is called the PAGE. Okay, so there is electrophoresis, gel electrophoresis, by which we separate the microbial proteins and transfer it, that's called blotted on a nitrocellulose membrane, which is incubated with patient serum. So this method is different from the previous method. We have blot, the result will be like blot, and we use gel electrophoresis in this method. Immunoblotting is a highly specific test which may be used to confirm the results of less specific tests such as ELISA. So this test is more specific than ELISA for detection of infection and microorganisms. The other test, what is called the complement fixation test, the patient serum is heat treated to inactivate complement and then add a specific antigen. Other tests, which is serological test for the microbiology detection, it is called the agglutination test, when the antigens are present on the surface of the particles, for example cells, latex particles, or microorganisms, or cross-linked with antibodies and visible clumping occurs. What is called this clumping called agglutination. There are different types of agglutination tests. We have direct agglutination test, or indirect, which is passive agglutination, or reverse passive agglutination. The other important test, which is called IGRA test for the Mycobacterium tuberculosis, as I said, it is one of the common infections in our country. The IGRA is antibody-independent specific immunological test depend on interferon gamma release assay. So this is the abbreviation of the IGRA. It is the interferon gamma release assay. Are being used increasingly to diagnose TB, which is the tuberculosis. The principle of the assay is that T lymphocytes of patients infected with Mycobacterium tuberculosis release interferon gamma. So the principle is the release of the interferon gamma from the T lymphocytes which are infected by the Mycobacterial tuberculosis. When they are exposed to Mycobacterium tuberculosis specific peptides. Okay, so the release of this interferon gamma from the lymphocytes. The absence of these peptides in bacilli, which is the calmette-guerin vaccine, result in IGRA test being more specific for the diagnosis of tuberculosis infection than the tuberculin test, because the latter may be positive as a result of previous BCG vaccination. This meaning a way of prevention of the tuberculosis, of course, by the BCG vaccine, which is given early in life in the neonate, of course. For the prevention, we give this BCG vaccine. The BCG vaccine has peptide of bacilli, and this peptide of bacilli process as this bacilli will give immunity. The body will form antibodies and will form a specific cell-mediated immunity against this type of bacilli, and this cell-mediated immunity will protect the person from developing Mycobacterial tuberculosis when he is exposed or she is exposed to this infection. The test previously was done for the diagnosis of tuberculosis. It's called tuberculin skin test. But this tuberculin skin test sometimes gives false positive, as the patient has TB, but he has immunity, not the TB itself. The IGRA test, which depends on the release of the interferon gamma from the T lymphocyte infected by the Mycobacterial tuberculosis, does not depend on this peptide of the bacilli, so it is more specific and gives accurate diagnosis than the previous skin test, tuberculin skin test. Now, so we have, as I said, multiple methods of investigation. Sometimes some of them are specific for detection of the microorganisms, and the other tests for detection of the serology of the patient by detection of the antigen of the microorganism or antibody that is formed against this microorganism. Now, while somebody may ask, what are the role of the imaging test in the diagnosis of the microorganisms? Of course, the imaging test and radiology plays a role in the diagnosis, but not like the detection by PCR, as I said, or amplification test, or by culture and sensitivity. Different modalities in the radiology are used for diagnosis of infection. For example, chest X-ray for the chest infection, whether tuberculosis or other infection, whether it is infection pathogenic or maybe even opportunistic infection. I mean by opportunistic infection actually is a type of infection that is not usually affect the person. For example, an immunocompromised patient may develop atypical mycobacterial infection or atypical pneumonia. Okay, sometimes there is what is called pneumocystis jirovecii infection. It is also opportunistic infection. So imaging tests may be used to give an idea about the infection by chest X-ray, for example, and also chest X-ray may diagnose the infection, give an idea whether it is bacterial, or just sometimes give an idea whether being fungal. But the final diagnosis by using other tests like the culture and sensitivity test. Ultrasound also is used to diagnose the abscess, for example, liver abscess, gynecological abscess, and so. With the development of high-resolution cross-sectional imaging, anatomic identification of the most areas of infection has become routine, and imaging the site of infection allows for diagnosis of treatment. This picture, for example, showing the gynecological problem as abscess seen in the ultrasound, as seen here. So localized collection of the pus can be seen by the ultrasound. This picture showing there is in the right upper lobe of the lung, there is infection with cavity, and the patient, if the patient has fever, sweating and loss of appetite, loss of weight, all these features are of Mycobacterium tuberculosis, and the patient at the same time has this feature in chest X-ray, so it gives an idea that there is high possibility of being TB of the chest, of the lung. And also the patient, by using the CT scan, we can give an idea about the infection. For example, these days the diagnosis of the COVID-19 can be done by using of the CT of the chest. In the chest X-ray, it's not very helping in the diagnosis, because it maybe give an idea. Usually in the basal area, usually there is ill-defined lesion and usually not associated with blood effusion, while here, as we see here, there is ground glass opacities is seen in the CT, give a more informative idea as being COVID-19 infection. Also, CT can be used for diagnosis of different infections in the body, like brain fungal infection, as we see here, and others things can be seen by CT test. Now, as conclusion, different types of microorganisms can affect human being, and some of them are normal flora to the body. Investigation, as I said, for diagnosis of infectious agent could be direct detection of the organism or indirectly. And the culture and sensitivity gives an idea about the antibiotic also that can be used